Hello everyone. Today we'll be doing a video presentation on the translation of mRNA into proteins in eukaryotic cells. Translation is the mRNA-guided process of creating proteins. In prokaryotic cells, which lack nuclei, transcription and translation happen simultaneously, so they happen at the same time. However, the process is a little bit different in eukaryotic cells. After the process of transcription, the mRNA strand leaves the nucleus and travels to the cytoplasm for translation.
Basically, what happens is mRNA travels to a ribosome where it will get translated into a polypeptide sequence, which makes up proteins. The process I will be describing is the first stage of translation, initiation. mRNA, or messenger RNA, is the product of transcription and contains a series of codons, which are combinations of 3 RNA nucleotides, as shown by the boxes. Each codon codes for a specific amino acid. This small ribosomal subunit binds to an mRNA molecule on its 5' end, as well as a tRNA molecule. tRNA, or transfer RNA, is an adapter molecule which adds the amino acid to the polypeptide chain. Each tRNA has an anticodon, which is complementary to a codon on the mRNA strand, and this anticodon specifies which amino acid it brings to the ribosome.
This is a diagram of the two-dimensional structure of tRNA. This yellow part is where the anticodon is located. In reality, it would actually look like this. The tRNA is actually all twisted up, and this yellow part is the anticodon. And here we can see how each amino acid is covalently bonded to its specific tRNA by an enzyme called amino acyl tRNA synthetase. The purple hexagon is an amino acid. In our diagram, it's the amino acid tyrosine. There is a unique synthetase for each tRNA and amino acid coupling. The amino acid tyrosine, for example, uses tyrosol tRNA synthetase.
So going back to this diagram, the tRNA molecule here is called the initiator tRNA, which binds to the P site of the mRNA strand, carrying the amino acid methionine coded by the start codon AUG. This is where the translation of the mRNA will start. Then the large ribosomal subunit is joined by the hydrolysis of GTP. This whole complex is known as the translation initiation complex.
Now that the initiation stage is complete, we're going to see how the polypeptide chain actually forms. This process is called elongation. There are 3 sites on the ribosome involved in this: the A site, the P site, and the E site. The ribosome reads in the 5-3 direction.
The A site is where the amino acid tRNA matches to its respective codon on the mRNA strand. The hydrolysis of GTP increases the accuracy and efficiency of the tRNA anticodon base pairing with the complementary mRNA codon. Then, the polypeptide chain held by the peptido-tRNA in the P site will form a peptide bond with the amino acid on the tRNA in the A site. Then, a process known as translocation occurs. It is also powered by the hydrolysis of GTP. The mRNA slides over one codon, and the tRNA in the P site will move to the E site, or the exit site, and is released. The tRNA now holding the polypeptide chain moves to the P site, making it a peptido-tRNA, and this makes room for the codon at the A site to be read. Accordingly, a new amino acid tRNA will bind to the codon there, and this process will continue to elongate the polypeptide chain.
Once the ribosome reads the stop codon in the A site, the process of elongation ends and the last stage of translation begins, and we call this termination. So here, the stop codon is UAG, and what happens is a release factor, which is a protein shaped like a tRNA, binds to the codon instead of a regular tRNA. The release factor promotes the hydrolysis between the bond of the last amino acid and the peptidyl tRNA. And so the bond is broken and the polypeptide chain is freed and leaves the ribosome. Finally, the ribosome is broken down into its subunits by the hydrolysis of 2 GTP molecules. And this completes the last stage of translation. All right, everyone. Thank you for watching.
So cringy.
Spicy content.